Well, let's do that. Let's go through. I will do this. I guess I'm going to do it by the order of people on my screen, and that's sort of on the order of people who came in. So, just some introductions, why you're interested in this, and an update on what you've been up to. And yeah, so yeah, I'll just start real briefly, and then Abhinav, and then Embre, and then Stella. So I am interested in this for livestock anaerobic digestion of small systems, and especially interested in the weatherization part of it, how that would work. I don't have much of an update except that we're finally getting the first livestock manure, 100% livestock manure digester new project in Vermont in 10 years. So I went and visited that. That was very interesting to see how they're setting it up. So that was great. I also learned that there's a defunct digester with three 7,000-gallon tanks underground that I might be able to salvage for a livestock project at a small sheep, well, not small, 50 milking sheep operation, 50 ewes. Yeah, those are some little updates from me. Yeah, yeah, 7,000.
Can we have a presentation on the new livestock digester?
You think it's of interest? We're talking about like a 300,000-gallon tank. This is a pretty standard thing, but I'd be glad to give a thing on it. I'm hip deep in a case study that I'm writing about it, so I'll have all the information. You really want it? Okay, I'll put that down as a possibility, and we'll see what other people feel. Okay, Abhinav, why you're interested, update.
Oh, well, anyway, hi, I'm Abhinav. I've been working on anaerobic digestion for the past, I guess, 10 years or so through my PhD and now as the environmental research engineer at the Freshwater Institute. Um, Most recent stuff that I have done is building the pilot scale digester at our facility, about 12 cubic meters, which I'll be talking about in the presentation. And I'm also doing some work with magnetic biochar for hydrogen sulfide control in anaerobic digesters.
All right, Embre, yeah.
Embre looks so excited.
Could be interesting. Okay.
Yeah.
Well, because this is something we always talk about as a closed loop for biochar, right? Like you make biochar with the poop and then you use it for the digester odor control, but nobody's ever shaken this out, right? So
there is a project at Dickinson College that they're actually doing this at full scale. We've been doing mostly lab scale work, and the magnetic biochar stuff is still lab scale, but Dan Cholkos at Penn State, he just is planning to start a new project with adding biochar directly into a full scale digester to see how it impacts the digestion process.
So this all connects back to Abhinav's work with Stephanie Lansing, who our person knows in what's our leader here in Vancouver? Simon Fraser, what's her name?
Jane.
Yes. So this is all tied together. These people all know each other and working on similar stuff. So yes, let's park that as a possible topic, this biochar thing, Dickinson College, and my new project. Okay. So possible items for the agenda. Okay. Okay.
More questions, just a couple more questions, please. Well, one question and one link. Jaime, who is also part of this group down in Colombia, has done a lot of work with biochar addition to psychophilic digesters. It's just put a paper out. So we should hook you guys up.
Yes, absolutely.
Yep. He and Lily, and I think Jaime Marti, right? So that whole Colombian contingent has been doing biochar addition for psychophilic digestion for a while.
For psychrophilic, you said?
Yeah, yeah. Huh. But it's psychrophilic insofar, I mean, it's like, it's the psychrophilic that's just on the shoulder of mesophilic, right? It's not cold. It's just it's Colombian digesters, so they don't add any heat, so they're kind of naturally warm, but they are not technically mesophilic, right?
Right.
You have to classify that a little bit.
Gotcha.
Um, and then what is magnetic biochar?
Uh, Essentially, it's a biochar magnetite composite that I've prepared in the lab. Um, Basically, what the process does, it disperses magnetite nanoparticles. So magnetite is an oxide of iron, and it
Mhm.
disperses magnetite nanoparticles on the biochar surface. I'd done some work with iron biochar, just regular iron biochar, that seemed to work well, but I started working with marine fish manure, and those produce a lot of hydrogen sulfide. And I'll talk about this in the presentation as well. And so I wanted to design a biochar that was capable of handling really high concentrations of hydrogen sulfide. So that's what I came up with, the magnetite biochar.
So it is not, it is to chemically enhance the sulfide removal. It is not to physically remove the biochar, kind of like, because when I heard magnet, I was like, oh, this is like a recovery mechanism.
It does, you can recover it as well. Yes, that's another.
So you can pull it out with a magnet.
Yep.
Oh my God,
Two
it's
reasons.
but I can't stand it. Okay, I'll shut up.
I
Okay.
think I'm done with my introduction.
Thank you. Hi, Merrick. And we're going around just giving little introductions here. Stella, please. You have an exhibit for us, I believe.
Yes, I do. I forgot about today's meeting, so I started my lab work. So I'm doing my lab work. So this is my BMP bottles, um, 22 degrees. I still have 37 to do. And I have results for hydrolytic activity and specific methanogenic activity, which is great. So yeah, I'm Stella. I am now officially, well, in fall, a PhD student under Dr. Jane Fowler. I'm studying psychophilic anaerobic digesters, specifically looking at the microorganisms within these digesters to see how they recover, how they respond to the cold, all that stuff. Right now, I am working on characterizing the inoculum. So we're doing the biomethane potential right now, or that's what I'm doing right now. So I'm basically measuring the methane through, here, I'll show you guys, sodium hydroxide. So here's sodium hydroxide. I did this because I'm like kind of multitasking, so I don't want it to drip into my samples. And the sodium hydroxide basically just scrubs away all of the carbon dioxide so I can get a methane measurement. And yeah, that's basically what I'm doing right now. Um, yeah.
What are we looking at, Stella? What is our methane production?
For 37, it's great. For 22, we were having like a really weird, I was having really weird findings where my blanks were producing more methane than my samples. But now my blanks are plateauing and my samples are increasing. So I think it was just because we didn't properly degas the inoculum before we actually created these batch tests. I think there were just so much residual organic matter for the microbes to like eat up that they were so happy. So yeah, so this test I'm hoping to run for like another 25, 30 days for the 22-degree ones. The 37-degree ones are super normal. I have an Excel sheet, I can tell you. But basically, I'm not going to pull it up, but the specific methanogenic activity for 37 degrees is double that of 22 degrees.
Hmm.
Yes. So I am kind of concerned for the huge reactor. I feel like
No,
it's going to take a long time.
we just have to give them time.
Yes, they need a lot of time.
We just have to give them some time.
Yeah.
Yeah. We need to run the 22 a lot longer.
Yeah. We were going to also run some at 12 degrees, but then we didn't have enough serum bottles, which is why we're only using 22 and 37. So that would have taken even longer. So that would be true psychrophilic. So right now, even 22 is not really, it's on the cusp of psychrophilic. but because it's just sitting in ambient temperature, there's a chance that the temperature is like fluctuating from like 18 to 23. So my readings are really weird. But I have to figure out.
In a water bath.
We other people in the lab are using all the other incubators, so I can't just take everything, unfortunately. Yeah.
Tornado.
I
All
would,
right, Ambray, you're going to have to dip into Brown and Caldwell's lab supplies and get her more bottles in a water bath.
yes, that'd be great. And more poo. We need more manure. Yeah.
Oh, well, that we need to talk to
Yeah, Jonathan and Devin.
maybe the contingent. Yeah.
Okay, so Merrick, can we go on to Merrick? We're doing introductions and just saying where you come from with your interest and what you're up to, any updates.
We're getting totally distracted by all the fun.
And yes, that too.
Hi, my name is Merrick. I just landed here in northern Norway. Again, I'm between Norway and Alaska. And I worked as a business developer here for a big biogas park, and then for a company here with fish waste going to biogas plants in Scandinavia. And now I'm in Alaska working with other energy stuff and work as a ski guide too. So I've just been joined the group and yeah, like to follow along. And I think that's it.
Yeah, I think that was our last presentation that you were the one to give our last presentation. Stella or Embray, what's up with the White Horse Project? Just briefly, where is that at? The what is it, 1,600-gallon tank?
Yeah, so I spoke to Jonathan like a few days ago. They have 99% of the materials to construct the tank. But the vendor didn't actually have all of the materials, so they're going through a second vendor right now to try to get everything. But their goal is to start construction on August 24th. And then we'll start running it and do
Good.
all the fun stuff. Yeah.
Yep.
Vendor stuff doesn't surprise me. I have had to deal with a lot of vendor issues, so I know what that feels like.
To me, the encouraging thing is that eventually there'll be a really good bill of materials and, where did the stuff come from? Like part numbers and everything, catalog numbers, everything. It will be really, that's a process in manufacturing anything is a good bill of materials that you really know what the stuff is. And yeah, so it's good stuff. All right, here we go. 1, 2.
Should I
Abhinav.
start? Okay.
Yes, please.
So I'll share my screen, the presentation screen. Let's see. Sure. Can everybody see this screen?
I got it.
Okay. So I already talked about who I am. And like I said, I work at the Freshwater Institute. I lead our waste to value program. We've looked at different strategies for valorizing fish manure. And one of our focus areas is anaerobic digestion because that's my bread and butter, and I've been doing this for the last 10 years. Okay, so a little bit more about my background. I am a chemical and environmental engineer by training. Completed my PhD from UMD, College Park in 2019, worked under Dr. Stephanie Lansing, mostly looking at animal and food waste, with anaerobic digestion being the focus. But I also did some work on pyrolysis, gasification, biochar, incineration, and life cycle impact assessment. So lots of different things. And since I finished up with my PhD, I started working at the Freshwater Institute, joined just before COVID, and have been working with fish manure ever since. So I'll talk a little bit about our facility first, before I kind of talk more about the anaerobic digestion work, just to give you a brief idea of where we are, what we do, what our core focus areas are. We have been located in Shepherdstown, West Virginia for about 30 years now. This is a drone shot of our facility that you can see on the slide. We are on a 100-acre farm, about an hour and a half away from both Washington, D.C. and Baltimore City. We've been at this location since 1998, 1999. This whole campus includes the original farmhouse, historic spring house and barn, 3 research buildings, 5 greenhouse bays, a pump house, and a small water tower. Our institute's goal is to support the viability and expansion of land-based aquaculture, primarily in the US and North America in general. And our program is funded by USDA ARS primarily. About 80% of our funding comes from USDA ARS, where we are basically in a cooperative agreement with them. But we also apply for other federal, non-federal, and international funding for research on recirculating aquaculture. So this is the spring house, which, like I said, provides us the water for our fish culture operations. I'll quickly go through some of these picture slides so I can get to the meat of the presentation quickly. This is our main building. It houses the majority of our offices, 2 labs, and a large research space, which we refer to as our wet lab. So this is the picture of this is one of the larger tanks, or the largest tank in our wet lab. This is what our grow out tank looks like. It's a semi-commercial scale culture tank, about 40,000 gallons. We get fish eggs from either USDA or our private partners. Once the fish eggs arrive, they're disinfected, put into an incubation system. Once the eggs absorb the yolk sacs, they're ponded into our fry systems. And then they move on to fingerling systems, and eventually they end up in our grow out systems. Embry, you have a question?
Well, I'm just curious, and I apologize for my profound ignorance, but
You're good.
I've never heard of this before, and I saw you had like NIFA and USDA. So is that where you primarily get your funding, is from USDA? And so are you kind of like a sub-research arm of them, or are you just a standalone institute that primarily, could you just say a little bit more about like what the
Yeah, of course. So we are essentially a extramural partner of USDA. We have a cooperative agreement with them that gets renewed every 5 years. The funding is appropriate through Congress. So essentially, because USDA ARS, that's the facility that is right down the road, don't have RAS systems, and we do have the RAS systems to conduct recirculating aquaculture research. They funnel the money to us, and most of that money goes on to do ARS related research, which is part of their 5-year plan. But that's mostly 80% of our funding where it comes from. The rest of the 20% comes from our other types of federal funding like NIFA, AFRI projects, sometimes even smaller grants, as well as contract research from the industry.
Oh,
does that help?
Sorry.
No, you're good. Yeah.
I didn't know if this particular project was a USD funded or if it's just like, okay. Yeah. Okay. Thank you.
So what I'll talk about today is I think it's pretty much all federally funded. Some of the hydrogen sulfide work is through the NIFA competitive grant stuff, but the rest of the pilot scale digester that we've built and everything else that goes along with it is funded by the ARS cooperative agreement.
All right. Sorry to interrupt. Keep going.
No worries. You're good. And please feel free to ask questions in between. Otherwise, I'll just keep on droning on.
Careful what you ask for.
You'll be fine. It's fine. We have 1.5 hours. We have plenty of time to talk. Like I said, we grow out the fish to market size in our grow out tanks, and we have been able to grow fish at biomass densities of up to 80 kilograms per cubic meter. And with the system that we have, we can recirculate more than 99% of the water. This is our another set of research scale systems. This is what we call our 6RAS. So these are basically 6 replicated smaller versions of the grow out tank, about 1,400 gallons. And this is what we typically use to do our replicated peer-reviewed research for recirculating aquaculture systems. We also have a newer building we call it our tank pad, which adds another 6 replicated systems, and it essentially expands our research capacity. So we have 12 replicated systems on our site to conduct replicated research. Additionally, we also have our water chemistry lab where we do a lot of our water quality testing. We are also designated a commercially certified lab by the West Virginia DEP. We have capabilities of testing, quantification of microbiological, nitrogen, phosphorus, organic solids content for aquaculture research. And we also do a lot of the testing for our facilities NPDES permits. Since our site has both RAS and flow-through systems for research, we are regulated as any point source discharge site. This is our pathology lab. We have this space for aquatic veterinarian, who's also our director of research, you see on the left. And it's also serving as our space, as my space for our waste to value projects. This is the most recent area that we've started to explore of precision aquaculture. Our most recent hire, Dr. Ranjan, has been working in this space and is utilizing machine learning and artificial intelligence to monitor fish underwater for health assessments, biomass assessments, so that we can, in the future, we can kind of analyze all of these things without having to handle the fish and stress them out. And this is our greenhouse. This is where most of my pilot scale work happens. We have our dewatering equipment in the greenhouse, as well as our full scale composter, as well as our pilot scale anaerobic digester. A quick like a primer on what recirculating aquaculture is for folks who are not as familiar with it. It is basically a closed containment method for fish culture. And this has been our area of research for the last 30 years or so. The water recirculates through the filtration units and is continuing being recycled and reused. It has a highly efficient filtration system that can support more biomass per volume than traditional aquaculture. Solid fish waste and uneaten feed is quickly pulled out of the water column, mechanically filtered, and then dewatered. As you can see in the bottom of this picture, we use a gravity thickening settler for dewatering for our wastewater. Dissolved nutrients like toxic ammonia is then kind of sent through our nitrification unit, which converts ammonia into nitrate, and then it is CO2 stripped out and oxygen is added back in before the water is sent back to the tanks for the fish. Yep.
You ever thought about recovering that nitrogen? Do you ever deploy?
I will talk about it a little bit, but yes, I've also started working on microalgae for nutrient recovery.
Oh my God.
Yeah, so
to go down.
We've just started, so it's very new for us, so we're still learning. But that's something that I've kind of thought about, like using just a heterotrophic, well, not heterotrophic, like a nitrification system for biofiltration. Why not use microalgae to remove your nitrogen, to remove your ammonia, as well as add oxygen into the system so that it reduces the need for added oxygen. And also, consume the CO2. I don't think I've seen anyone do this at scale except this one farm or this one company that is trying to do this for shrimp. I know the guy is based in Ohio. And they're the only ones I know who's trying to do this for shrimp, trying to basically take the sand-filled biofilter out and put it a microalgal biofilter.
I'll talk to Craig. Craig just does at University of Iowa does a ton of stuff with algae. So
Okay.
I put his stuff in the chat.
I'm not being able to see the chat for some reason. Maybe that's why I've been missing on the
I'm sorry.
notifications. Okay, that makes it easier. All right. So
I just want to welcome Judith. Hi, Judith. You kind of missed introductions, but we're underway.
Yeah,
And
sorry.
feel free to chime in.
Yeah, we'll talk after.
So, and these are basically all our research areas. This is the core research areas that we tend to focus on. So that's basically our background of our institute and what we do. I'll now move on to our waste to value stuff. So this is my program. This is our waste to value program. We started this in 2020 when I joined the Freshwater. We basically started taking solid waste from RAS systems and trying to make it a source of added revenue for farmers. We've looked at anaerobic digestion. We've looked at composting. We've also looked at mortalities or processing fish processing waste for anaerobic digestion as well as composting. So this is not new information. Everybody knows that waste in general, organic waste in general from animals tends to be rich in organic matter, nutrients like nitrogen, phosphorus, but unfortunately, in a lot of RAS facilities, especially in the US, I know in Europe they are a lot better at handling waste substrates. But in the US, a lot of the new facilities don't really think about how to deal with the waste. The focus has always been on more discharge or like basic treatments, but not repurposing the waste for added revenue. And again, it makes sense because recirculating aquaculture or land-based aquaculture is relatively recent compared to terrestrial animal farming systems that have been around for a long period of time. So a lot of those technologies, waste treatment technologies, have been developed really well for those waste substrates, but not as much for aquaculture waste streams, which is kind of our goal to essentially adapt some of those technologies and bring them into land-based aquaculture. To give you some more context of how much waste these systems can produce, the standard rule of thumb is for every kilogram of feed you feed the fish, about 25% is excreted out as solid dry matter. Basically, 250 grams of TSS per kilogram of feed is the rule of thumb. And if you convert it to like annual production, like if a fish farm is producing 5,000 metric tons of fish a year, they will produce 4 metric tons of dry waste per day, which is a lot. But then you have to understand that they don't produce the dry waste. It's not dewatered to 100% dry matter. It's only dewatered to maybe 10% dry matter. So you're looking at 40 metric tons of waste every day, which is a lot of waste that needs to be dealt with. But unfortunately, not a lot of farmers are doing anything with this waste. And typically farms in the US, the largest farm in the US, Atlantic Sapphire, they have a capacity of, I think, 10,000 metric tons annually, but they haven't been able to make it to that level yet. They're probably, I think, at 8,000 or something like that right now. But it's still a lot. So a lot of the waste that they produce is sent to landfills, or the solid waste that they produce is being sent to landfills right now. So for our research, like I said, we've been looking at all these different types of things. Anaerobic digestion has obviously been our focus since that's my area of expertise. And like I said, we've done composting and we've also started working on microalgae just now. And we'll be working on microalgae at least for the next 4 years. I will not talk about what anaerobic digestion is because everybody knows what it is. But just a fun fact that I always include this slide in my presentations because whenever I go to aquaculture conferences, it's very rare for me to find people who do anaerobic digestion. So it's always good to include this so that people in my presentations can understand what I'm talking about. So a lot of the inspiration for what we've done since I joined Freshwater has been inspired by this review paper that I wrote back in 2022, essentially highlighting all the challenges that um, that are prevalent in aquaculture waste. That before it can be digested in an AD system. The first one is a dilute waste stream. If you don't dewater your waste, you are looking at solids content of 0.1 to 0.5% TS, which is quite low. Then you also have that low carbon to nitrogen ratio issues. Typically, it's less than 10. From our experience, we've seen anywhere between 5 to 7 CN ratio, so very much on the lower side. Then you also have high salinity, and as a result of that, high hydrogen sulfide concentrations from marine facilities that use salt water for growing the fish. We are primarily freshwater, so that salinity issue doesn't really impact us as much, but we have collaborators who do, full strength seawater or brackish water, and then their sludges mixed with a lot of salt. The high lipid content is primarily associated with processing wastes, and we've done some work on that as well. So in our starting, yes, Embry.
Why is the sulfide so high in this particular waste?
Because it's got a lot of sulfates in it. Marine, like seawater, has a lot of dissolved sulfates in it. The second reason is you also have proteins in a lot of excreted proteins from the fish manure. We've seen about 35% on a dry matter basis proteins in the fish manure. And that is another reason why the CN ratio is really low. So yeah, both of those factors directly lead to the high hydrogen sulfide concentrations. And I'll tell you what the high hydrogen sulfide concentrations are. It'll blow your mind. It's a little bit later in the presentation. So this was the first lab scale study that we did, looking at essentially thickening the waste from 1.5% dry matter solids to about 3.5% dry matter solids. And overall, we saw about a 23% increase in the methane production as we increased the dry matter content. So that was beneficial. And we did some follow-up studies with increasing it even further, and I'll talk about it a little bit later. In our follow-up study, we kind of looked at how does a diet impact the biogas production how does the diet impact the waste characteristics as well as the biogas production? And in the study, we actually tested 2 different diets, but with both of the diets having similar crude protein and crude fat levels. But we found that the waste was completely different. The waste characteristics were completely different. One had a much higher crude fat content, but very similar crude protein content to the other one. And as expected, the waste that had a higher crude fat content led to a higher methane production in the system, as well as a higher COD removal from the fats. This is something that we haven't published yet. I'm still on my desk waiting to be published. But basically, we kind of started looking into what happens if we increase the concentration even further. So we went from 5.5% solids concentration to about 13% solids concentration, even though the total methane production was kind of similar across treatments. But if you start looking at the data closely, you'll see that there was a much longer lag phase or slower rate of production once we went above that 5.5% solids concentration. So for minimizing inhibition, at least during startups, 3.5 to 5.5% may be more appropriate. But then the microbes do tend to acclimate because we've done microbiome analysis on the systems, and I'll show you that a little bit later. On the right-hand side, this is the work we did with. We actually combined or co-digested fish manure with fish processing waste, so mostly the guts, to essentially kind of balance out the CN ratio. And again, there was a lot longer lag phase when you had all the fats included in the system. But eventually, the co-digested treatments with the fish guts led to about a 50% increase, a maximum 50% increase compared to just raw fish sludge. Again, you needed time to acclimate. You can kind of see on the x-axis it took us 60 days to get to that level. But if it is just fish sludge, there was barely a lag phase compared to all the other treatments with the fish sludge. And you can actually see the fats kind of floating on the surface and eventually disappearing over the 60 days. That was kind of cool to see. And this was all very evident in the microbiome as well, like how the microbiome with the fatty substrates essentially shifted to include more bacterial species that were capable of breaking down long-chain fatty acids. So yeah, just the microbiome stuff. I'll quickly go over it. So basically, you can kind of see in the inoculum, you had a lot of metanocyta, which is an acetoclastic methanogen, and doesn't handle high ammonia concentrations really well. This is from the study where we changed the solids concentration from 5.5 to 13%. And you can kind of see the increasing relative abundance of the methanosarcina, which is more tolerant of high ammonia concentrations and can have both and can switch between pathways, acetoclastic as well as hydrogenotrophic. So we kind of saw that happening in this system as well, which was what should have happened in the first place. But yes, it was really nice to see that, that shift from the inoculum to the treatments. Same way, when we checked the microbiome of when we co-digested the fish guts with the fish manure, different microbiome, but like I said, you had a lot of methanosarcina, which is again highly tolerant of inhibitory conditions once they get acclimated to the system, and we had a lot of hydrogenotrophic methanocullius as well, and a lot of synergicities, which are capable of handling these fatty substrates. Okay, so this is the part where I'll have to maybe go through a little bit quickly, but it is the part with the hydrogen sulfide stuff. Basically, the high salinity and high hydrogen sulfide can lead to a lot of issues. You're all familiar with hydrogen sulfide related problems. This is a picture on the left of the part of the screen. This is a picture of a generator from a farm in Maryland. And this is the first farm I visited when I was doing my PhD. And you can kind of see how badly corroded this is. And they would be, there was a lot of downtime for this generator as well because they were constantly changing parts of the generator because of the high hydrogen sulfide concentrations. There are market available systems for hydrogen sulfide treatment. But my interest has always been, what else can we do? Is there an easier way to do this? And that's why I kind of started looking into adding biochar directly into a digester, as well as comparing it to having biochar as a scrubbing system. So I've always done the research in that way, like adding biochar directly in a digester, as well as using it as a scrubber. And this work actually kind of connected me to Alex in the first place, back in, I think, 2018. Or something like that. If you're not familiar with biochar, it's basically it's an umbrella term that people use to talk about any biomass that has been subjected to thermal degradation under a no oxygen and low oxygen environment. All biochars are different. It's typically used for soil amendment, moisture retention, environmental remediation, but its application in anaerobic digestion is very recent. We did a lot of the initial work with some of the lab scale stuff, and I'm happy to see that Penn State is starting to look into actually adding the digester at full scale to see how it impacts a full scale system. So this is the modification process, like I said, for the magnetic biochar that we used. It's a published method, but I don't think people have used magnetic biochar for hydrogen sulfide control in digesters. They've seen it as an effect, but for us, the goal was to focus on the hydrogen sulfide controlling aspects, like basically bring down hydrogen sulfide from really high concentrations to almost negligible concentrations so that the biogas can be used directly in a boiler or a generator. So this is the modification process that we followed. And this is what the biochar looks like under a scanning electron microscope. So on the left, you can see it's a pristine biochar particle with no magnetite impregnation. On the right, this is what the biochar particle looks like after the process. And when you zoom in even further, you're starting to see these nanoparticles agglomerated on the biochar surface. So it basically creates a more uniform distribution of the magnetite nanoparticles. So it enhances the surface area for better reaction with the hydrogen sulfide. We've done some X-ray diffraction just to prove that this is magnetite, and some really clear peaks that show that this was magnetite being deposited on the biochar surface. And this is the part. So hydrogen sulfide from these marine systems, oops, I think I need to go back. Oh, hydrogen sulfide from these marine systems, you can see on the right-hand side, has crossed, at least in our studies, 25,000 parts per million. In any dairy manure systems, I've never seen anything beyond 5 to 7,000. Yes, I know. It's absurd. So
Just a second.
yeah.
I can't hear Embre, but I can just read the body language. But whatever. Go ahead. What are you saying?
Well, but even a high protein poop doesn't explain that. So is this really like the marine sulfate intrusion? If that's the majority of it.
Yes, I would say so, yes. And this has been seen by other researchers from Europe as well. I have a close colleague who works at DTU, and they have seen concentrations exceeding 15,000 parts per million from their marine waste systems. So this is not uncommon if you're subjecting salty fish manure to anaerobic digestion systems. However, if you look on the left-hand side, there's no inhibition, which was interesting for us to see because, or not interesting, because it was actually expected, because we were using an inoculum that was developed by our colleagues at the University of Maryland, Baltimore County, that they isolated the several strains from marine sediments that have these halophilic methanogens as well as bacteria. They enrich those species in that inoculum. I believe it's patented as well. And we used that. And as a result, we did not have any lag phase. You were getting to almost like 80, 90% methane production within 10 days. And from what Stella was talking about, it made me wonder if there are ways to isolate and enrich methanogens from cold climates to enhance the methane production.
There are, and one of our cohort, Tom Curtis, does that. And Jaime.
Has it been very successful, or is it still? It has been. Okay, good. Yeah. So.
Yeah, get a hold of Tom Curtis at Newcastle. He goes to Svalbard and collects like anaerobic lake sediments. And then goes.
Yeah. Fun. Yeah. Work with fun. Nice. So,
I don't think Pernilla does that. Pernilla doesn't do a lot of microbiome stuff, right, Judith?
I'm sorry?
Pernilla doesn't have her own like enhanced psychrophilic methanogen culture.
Not that I know of, but maybe something new has come up. No, I've never heard of one.
No, sorry.
I'm sorry. I'll shut up.
No, you're good.
We also, at the very beginning of this group, I've always kind of kept her in the back of my mind. But at the very beginning of this group, we had a microbiologist who worked for the U.S. Army Corps Center for Research and Development, Cold Climate, who was a psychrophilic microbiologist. And so she might also, Dr. Robin Barbado, might have some experience there. I can send you her names. And you can't see the chat, otherwise I would send you the chat.
Yeah. Okay. So again, in this graph, you can see it was, peaked at 25,000 ppm in the control, but biochar by itself was able to bring it down by about 50 to 60%, but it kind of lost its efficiency over time, or at least 2 of the biochar samples lost their efficiency over time. But even then, it was not able to fully remove the hydrogen sulfide concentrations down to what's required for stable operations in generators and boilers. But if you look at the line at the bottom, all 3 magnetic biochars were able to bring it down to below 100 ppm throughout the experiment. Initially, I think on day 2, it kind of peaked at about 150 to 400 ppm, but after that, it was all the way down to less than 100 ppm for all the treatments. So really positive results from this experiment. But some of this stuff worries me, that the biochar loses, the unmodified biochar losing its effectivity, which makes me think that the biofilms forming on the biochar surface might restrict access for the hydrogen sulfide molecules to react with the biochar over time. But we don't know that yet. I have not looked into it too deeply, but that's the theory that I have right now. Again, microbiome, the difference in the microbiome, very interesting differences because you see a very different microbiome on the biochar surface versus a sludge without the biochar. And again, that makes sense because the biochar houses a lot more of those electroactive species that can get involved in something called DIET, or direct interspecies electron transfer. I'm not going to go too deep into it, but essentially it helps enhance methane production, or the rate of methane production in most recent research. Okay, so then we also did some comparison with the same magnetic biochar as an external scrubbing system instead of adding it directly into a digester. We did some of this work in our lab scale units. We built this in-house. And again, you can kind of see how much higher the capacity is for the standard biochars versus the magnetic biochars, about a 11 to 25-fold increase in magnetic biochars compared to the regular biochars. So that was very promising as well. I just finished writing the paper for all of this stuff, and I'm hoping to get this published by the end of this year. Okay, so some cool. Of course, yes. I'm sure Alex will be on it before I am, because he keeps track of all this, the hydrogen sulfide and biochar stuff. There's also some more cool pictures. So this is what on the left you see what the biochar looks like before the reaction happens, and this is what it looks like after the reaction. You can kind of see how the particles change. It was like these small nanoparticles that now more like look flaky crystals. And this is basically another version of iron that is very similar to rust. So we don't know, we haven't tested it for, if this biochar can be regenerated and reused for hydrogen sulfide removal, but that's something that I want to explore further to see like if this magnetic biochar can be reused multiple times for hydrogen sulfide removal.
I'm sorry, but this is kind of like using an iron sponge for sulfide removal, right?
More or less, yes. But this is, I think, might be better because it has a lot more surface area as well as the nanoparticles. I haven't compared this to the iron sponge that is commercially available, but I don't know what these capacities are for iron sponge. But if we could compare, yeah.
You can regenerate iron sponges. Right?
To a certain extent, right? It's usually like about 30% capacity is lost every time, which might be the same here, because in this, you can kind of see the SEM picture show you there's like a lot of sulfur deposition on the biochar surface. There's the carbon, there's some oxygen, but iron and sulfur, like there's some spots where the sulfur is more concentrated. And I think this is mostly elemental sulfur because we've done some XRD analysis and it shows crystalline elemental sulfur forming, which is likely to basically block the pores and reduce its capacity for regeneration. But I think it can still, like iron sponge, like you said, like we might be able to use it a couple extra times, but I want to do some more research on this to see how often it can be regenerated and recycled. Okay, so main event. This is our pilot scale anaerobic digester. So you can kind of see What it looks like on the left, this is our in-house design. We pretty much did it ourselves, building it using stuff that we already had and also added some new stuff. And on the right, you see this is our control panel, which allows us to automate everything, because managing the digester without automation was proving to be very challenging for the first year, especially when it comes to maintaining temperature of the digester. So yeah, we decided to add this control panel this year, and it's made things a lot easier for management purposes. This was the design that we did in-house. So essentially, sludge from our gravity thickening settlers is pumped into this equalization tank. I kind of call it a premix tank, which allows us to basically combine it with other substrates if you wanted to co-digest the material. From this, there is a submersible pump in this system, in this tank, and that pump essentially pumps the sludge into this main tank, which is our digester tank. This is 8 feet deep and 8 feet in diameter, which is about 11.5 cubic meters. And we have about a 10 to 12% headspace in the system. And as soon as the fresh sludge comes in, there's an overflow pipe that basically allows the sludge to overflow into this digestate tank, which is then pumped back into our holding cells before someone else can come in and take it away. We are not allowed to land apply this because we are classified as an industrial waste generator. So we need a proper permit to land apply this. So that causes a lot of issues for us. So that as a result, we've just been asking someone else, a sludge hauling company, to come in and land apply it somewhere else where they do have a permit. These are the control drawing for the system. Again, all of the sensors and the mechanical equipment is connected to the control panel so that we can automate everything based on sensor readings and everything else. Also, we have alarms to make sure that, if there are safety considerations, if there's any safety issues, it alarms out through the control panel. So when we started this project, we had a lot of uncertainties because this is a novel waste. And again, a lot of the projects like this don't really, or at least I haven't been able to find a lot of projects at this scale. We are also kind of restricted because we had to design the digester inside rather than outside. So there were a lot of, we had to make sure that it was done safely for, operator safety, facility safety, looking at hydrogen sulfide issues. How do we heat the digester properly? We had to deal with flammable gas. None of my colleagues, because we don't really work in this space. I was the first one to kind of come in and work in this space. So there was a lot of, fear around dealing with flammable gas, especially flammable gas with hydrogen sulfide. And that makes sense, right? We have heard so many stories about farmers, or the kids playing around, manure fields, falling in and dying off and stuff like that. Nobody wants negative press stuff here, especially we are a nonprofit because we're getting USDA funding. We have to do things very safely. And obviously it makes sense to do it safely. We don't want any employees getting hurt. So we spent a lot of time figuring out how to do this safely and within budget. Because again, you can add a lot of things, but that adds a lot of cost to your system. So we have a sensor phone, basically that is connected to to the control panel and all our sensors in the greenhouse bay. So the greenhouse bay is where we keep our digester. So we have these McCurko sensors that are basically ambient monitors for hydrogen sulfide, methane, and carbon dioxide. And these are all connected to the sensor phone. We also have a portable detector that detects methane, carbon dioxide, hydrogen sulfide, and ammonia. So when the operator goes into to that space, they always have an idea of if there is any localized hotspots where if there's a leak or something like that, as well as a fire extinguisher just in case there is, a fire hazard or anything like that. The other thing to note is that we also have a ventilation system in that bay. So there's a switch on the outside that any operator that goes in can turn on the switch so that the ventilation allows for airflow inside that bay before someone goes in to keep everything to prevent any toxic gas buildup. We're still looking for more. We still keep adding things, or at least looking into more options to make things very safe. And one of the things we started looking into is using explosion-proof equipment for our gas handling systems.
I just wanted to ask, Embre and Stella, has this been, we talked about this issue at the Whitehorse Project a little bit, like codes and safety and all that stuff. Is that still a big issue there, or has that kind of been settled? Did you ever find a flare?
So I think we decided that we didn't want a flare for that project, Alex, because I think that the flares
Yes.
we were aware of needed a natural gas pilot connection. So we didn't want it. So I think they're just going to go out and like periodically. But I think John was going to talk to the insurance people and the code people, and I haven't heard back, Stella, if he's talked to them or not.
Yeah.
Yep.
So he's talked to them. I think it's pending right now. And then he said that, let me just check something because I have it written in my safety thing. I think it is supposed to be approved.
But maybe that's something, Abhinav, we can share with you or vice versa. Do you have a flare?
I will talk about the flare that we've
Okay. Sounds
looked
good.
at. And I think it might be able to solve that natural gas challenge that you might have, but it's in a later slide.
Good.
Yeah.
So yeah, safety, a lot of safety considerations. We're still adding to it. We're still improving on it. We have started working on an emergency action plan in case something bad happens. And we're constantly attending safety, manure safety workshops and stuff like that, just to make sure that we know everything about handling manure systems, as well as hydrogen sulfide issues and flammable gas issues. So these are the type equipment that we use. Essentially, we have our settler thickener, we have our tanks, pumps, agitators, heat exchanger, boiler, all the different sensors. We also have a gas analyzer that measures the quality of the gas continuously, so that it's required for our research, so that we can track the quality of the gas, as well as plumbing and gas storage. So I'll show you pictures of everything that we have for our system. So this is our gravity thickening settler on the left. It's about a 1,300 gallon system. We have 3 of these for our facility, and 2 of those tanks are plumbed into our inlet, and the 1 is plumbed to our outlet, or a digestate. We use this air-operated diaphragm pump to suck out the sludge from the bottom of these dewatering systems. This is a Sandpiper Beast. I love the name. Which is supposed to be a non-clogging pump. It has worked pretty well for us so far, and it's highly capable of transferring, sludge that is 10 to 15% solids into our equalization tank. We've had some challenges, but overall, it's worked pretty well for us. We've had this pump for about 3 to 4 years, 3 years now. So this is what the system looked like. We actually had some restrictions on what we could do with our tanks, because we are using tanks that are 20 plus years old. So when I joined the Freshwater Institute, my boss told me, hey, here are some old membrane biological reactor tanks, retrofit into an anaerobic digester. So I didn't have the flexibility of adding new tanks or anything like that. I just had to retrofit whatever we had to kind of transform into an anaerobic digestion system. So I tried my best to do it. Seems to be working fine for now. But essentially, we just removed everything and covered up, changed the design, added new pipes and everything, and then this is what the finished digester system looks like. We covered it with a geomembrane liner. Got some help from, I think, IEC covers. They kind of provided us with some suggestions on how to do it properly, how to cover the digester tank properly. And if anyone's interested, I can send that information to you. We are using these Creeman chopper pumps that are capable of handling, fibrous manure solids. This is on the left. This is our submersible pump that is installed in our equalization tank and our digestate tank. And this is our bigger 3 horsepower pump that is recirculating the sludge through our heat exchanger, as well as it helps keep everything well mixed so that the sludge doesn't settle down doesn't settle down and creates a cake layer at the very bottom. We also have a bypass loop just in case the heat exchanger needs to be, fixed or repaired or whatever, so that we can keep running operations if there is some kind of a downtime on the heat exchanger. We are for agitation in our premix tank and our digestate tank. We are using these 20-year-old Cheminair Nord agitation units. It has an axial flow mixer. Again, basically because we are short on money, just trying to reuse whatever we had to make these systems work. And thankfully, these systems are still pretty good and are working just fine for us. This is our heat exchanger. It is a standard, shell and tube heat exchanger, which we got from this company called Comax. They're based in California. They specialize in sludge heat exchangers. They have baffles inside the tube side. That's static elements that help keep the sludge well mixed. There is a pressure drop about 1.5 psi across the heat exchanger, and the delta T for a single pass is about 2 Fahrenheit when we set the water temperature, the hot water temperature, about 155 degrees Fahrenheit. It is counter-current flow, and we also looked at other types of heat exchanger units. There were systems that could, there were coils. There are systems that had like coils inside the digester tank. We did not like that option because if there was some kind of a problem in those coils, we would have to open up the tank and then fix it. So we wanted to have something external. So just in case that we needed to repair something, it was more easily accessible. So that's what we ended up going with. It seems to be working really well. We haven't had any clogging issues with this. Plus there are ports for temperature sensors in the heat exchanger that allows us to kind of monitor what the temperature is going in and out. This is the propane boiler that we're using right now for maintaining temperature inside the digester. We thought about using biogas boilers, but we couldn't find anything at this scale. So we just use a combi boiler just typically used in home systems. And we were using an ethylene glycol and water mix to heat up for heating up our sludge. We looked into what we could do to modify existing natural gas boilers, but because that required manual modification, it was not something that you could buy off the shelf. There were safety implications that most of our employees were not comfortable with. So we decided to just go ahead with a propane boiler for now. But eventually, if we figure out how to do this safely, we might look into having a primary boiler that is generating heat using biogas, with the propane boiler acting as a backup. The other thing for this boiler system was, even though the boiler itself is not that expensive, we did not have the expertise to install everything or the piping everything in-house. So this was one of our most expensive installations for this project. The entire thing with all the piping and the pumps and everything was about 30 grand. So this was one of the most expensive part of our projects.
How many BTU per hour or kW is that?
120,000 BTUs. And that is sufficient to actually keep our digester warm, as well as it also sends hot water to a hydronic heater, which is able to keep the entire bay warm during the winters. We've had some close shaves this past winter when temperatures dropped really low, because we were not expecting it. But overall, because we have heating systems in the other bays as well, if you keep the door open, we haven't had any freezing issues inside the bay. So these are all our sensors that we use. Most of the sensors that we've gotten are from Automation Direct. We have all these temperature sensors, pressure transducers. We also have these meters from EKM metering, which I've also used for my PhD. They're pretty good at measuring gas flows, so which can help keep us track of how much biogas we are generating. For our flow meter, we are using a George Fisher flow meter. Yes, yeah, I saw that heat exchanger for at the Penn State farm as well, but I don't know how much they cost.
Yeah, it's probably
Uh,
for bigger flows.
yeah.
Do you have like a whole bill of materials?
I can get you everything, I think, except the tanks, because I don't have anything about for
Yeah,
that
no,
for
that's
that.
okay. I just like
But the
it's
rest
one
of
of
the
those things
stuff,
where you can lose track of what you actually
yes,
installed unless you keep writing it down.
yes, I do have everything else with me because I pretty much sourced everything by myself. Um, so, and this is the George Fisher flow meter. It's a cool flow meter because you can actually connect it to your phone and you can kind of track the volume of sludge that you've added to the system and how much volume you've added over time. So it's a pretty useful, nifty tool, which I kind of like. But there are some challenges because sometimes it does send a false reading to the display. I don't know why that happens, but flushing the system kind of helps clear it out. So I'm not really sure exactly why that's happening, if it's an electrical issue or if it's like a stuffed biofilm that is kind of causing it to read weird values. We'll have to figure that out. This is our MRU SWG 100 biocompact biogas analyzer. We've been using this for about 2 to 3 years now, when, not 2 years, 2 years actually. It's working pretty well for us. You can kind of see what our biogas quality is right now. About 75% methane, 20%, 25%, 22% carbon dioxide. Not enough hydrogen sulfide right now because we're not feeding it actively. Because I am going to be on vacation come in the next couple of weeks. So we are just kind of slowing things down. And when I'm back, we'll start feeding it again with aquaculture waste.
What do you mean, not enough hydrogen sulfide?
It's only 1,000. I told you 25,000. That's
And you
crazy
want
stuff.
more so that you can figure out how to mitigate it? Or what do you mean?
For me, anything below 1,000 is not that much.
Yeah, I get it. So,
Yeah.
all right. It's not a good thing. You're still thinking that as a
Right,
bad
right,
thing. Right. Okay.
right. But in terms of like numbers that I've seen on different farms,
Yeah.
this is on the lower end of things. But we do see, like, once we actively start feeding our digester, we do see it spike up to 2,500, 3,000, 4,000 ppm. Because we haven't been feeding right now, it's kind of just gone down quite a bit. We do expect, production of about 5 to 10 cubic meters of gas when we are at full capacity. Right now, I think we maxed out at about 1.5 cubic meters a day, because again, we are feeding very dilute sludge, like 0.5% solids, because we did not have enough fish on site. But next year, when we have more fish on site, that's when we'll be starting ramping up this project, as well as doing actual conducting research trials on modifying the organic loading rates, temperature changes, and a couple of other HRT as well. I think playing around with some of these things to see what's the optimal conditions. Yeah.
Highest percent methane that you did?
75.
That is the highest.
Yeah, we've gotten 75% methane.
Same
Or I
as
mean,
that-
around that, because
Yeah.
it's a little bit higher than what we see when we check it on the GC, but it's around that 75% mark. And I think it's mostly because fish waste also contains some levels of fat. And if you use fatty waste, you tend to have a higher methane concentration. We are using, again, this is what we could find. You're using these push-in biogas bags, not the best option because they don't handle well under certain conditions, because we've seen some of the black bags, lose pressure because they had like micro holes in it, or like some animal chewed through the system, chewed through the bags, and all the gas leaked out. So we're still trying to figure out a better gas storage system, but we haven't found that yet. But as of now, we have about capacity for 8 cubic meters of storage right now. But eventually, when we start producing, 5 to 10 cubic meters a day, we would need to, flare it off constantly because we are not really planning on using this energy, at least as of right now. So,
Well, eventually, right?
eventually, yes. This is our control panel. I love this thing a lot because it took us a lot of work to kind of get this thing because, when Stella was talking about vendors and stuff, this is the issue that we were having, like finding the right vendor who was responsive, had good feedback from their other clients, because we actually had a very bad issue or a very bad experience with one of the vendors that we we're trying to work with. We worked with them for about a year, and initially they quoted us a price. When it came to us sending them the PO for the actual, starting the construction of the panel, they said, "Oh, your specifications, we didn't see ABCD, so now the price is going to be like twice as much." And that was like, well, that's not professional. We sent them a proper engineering document with all the specifications we needed. No word from them. They were like, "Oh, this is fine." And then eventually, right at the end of the negotiation process, they were like, "Oh yeah, it's going to be more expensive now." So we basically had to stop that, restart the process, find a new vendor, which took me, I don't know, another 3 to 4 months. Again, going through the quotation process, getting multiple quotes. So it was a huge headache on my part and actually delayed the project by about a year. But eventually we found this really great controls manufacturer in Syracuse, New York. They're called NE Controls. Have a pretty good history. The technician they sent to do the field training for us is also very knowledgeable, and they designed a really cool system for a very reasonable price compared to all the other options that we checked. We can basically control the amount of sludge that we add every day, how often we feed the system, how often we recirculate the sludge through the heat exchanger. It also allows us to communicate with the boiler to send heat or hot water to the heat exchanger whenever we need. And it also has some cool other features where it allows us to make sure that the slug of sludge inside the heat exchanger doesn't heat up too much. Because if there's hot water in the system, if there's sludge just sitting there, it might heat up too much and start to form cakes, like sludge cakes inside the heat exchanger. So you wanted to make sure there was enough flow through the system to keep things cleared before the temperatures cool down to more appropriate levels. So we can do all of that stuff using the control panel as well. So yeah, it's been helping us automate our systems pretty well. So it requires a lot less labor, because before we had this control panel, we were manually doing a lot of these different things. The other thing we had challenges with was our facility only gets single phase power, and a lot of the pumps that we were using utilizes 3-phase power. And so we had to get VFDs for all of these systems, which added to the cost, but it made it pretty easy for us to communicate with the control panel, so we can control everything using this control panel. So flare. Okay, so this is what the flare that we're using right now, the simplest thing. So you can kind of see how the biogas is burning right now. Again, clean, you don't really see it, but this is temporary because we're not producing too much gas. But like I said, eventually we'll have to switch to a bigger flare. So this is the one that we found. Yeah.
So I know home biogas that as a system, right? Like,
yeah. Mm-hmm.
You just buy that by itself?
Yes. It's about $125. You can buy it separately. Yes. Yeah. So it's about 4 kilowatts per burner. But it's still pretty slow. You're only burning off about a cubic meter of gas per 45 minutes to an hour. So it's relatively slow. And I had to, because we are, again, very safety conscious, so we have to make sure that someone is sitting there while the biogas is flaring off, because we don't want an open flame. And I spent 4 to 5 hours sometimes in our greenhouse making sure that nothing bad happened. So that was not obviously sustainable in the long run. So we kind of looked into solar powered flares. And this is the one that we found. It's by a company called EPG. It's a passive vent flare. It has solar powered batteries that I think can keep the spark on continuously. It's about $4,500, but they can handle It's too expensive. But they can handle like low flow rates, like about 2 CFMs is I think the lowest flow rate that they can handle. But this is something that we might consider next year for our FY27 cycle where we'll have new money coming in. So then we might be able to buy this for our system. But right now we're just using this home biogas, and seems to be working fine. It seems, it does the job. But again, if you're producing a lot more gas, this does not seem to be a good fit. But if you're producing less than a cubic meter a day, maybe this is a more reasonable option because it's very cheap. And yeah, so this is our final pilot scale digester setup. You can kind of see the geomembrane liner being inflated because that's what it looks like when the biogas is, the bugs are happy and biogas is being produced. And on the right, this is our tiny little microalgae setup that we just started working on last week. So we'll be working on some more microalgae research in the near future. So this is what our current research looks like at Freshwater Institute. And yeah, thanks to our funders for all this work, for all the funding to kind of do all this work. And I can, I guess, take questions. I don't think I have much time, but yeah, I can take questions.
I think I've peppered you with a lot of questions.
Happy to answer more.
So I have a couple of questions. So you are part of like a resource, kind of like a resource optimization, resource recovery division, right?
So yeah, that's my program. Yes.
So
converting.
When I see all that sulfur, I'm like,
can you do anything with it?
Yeah, totally. Like there's the biological recovery where you can just precipitate out elemental sulfur and use it for whatever, right? Agriculture or horticulture or keeping fleas off your yard or whatever. So I have a question about like, once you like, what if you guys have any plans for doing additional nitrogen recovery, like we talked about, or elemental sulfur recovery, or just anything like that? Because it seems like this group loves to talk about anaerobic digestion and gas recovery, but there's so much richness in this group that could be utilized. So like, I'm just asking about other stuff, like a little bit outside this purview, but again, like there's so much interesting stuff going on in this digester and in the coop itself.
Right. So recently, Alex got me in touch with a researcher who, I think, Alex, what was her name again? Because I think we were talking about trying to use the biochar after it has captured the hydrogen sulfide as a soil amendment to see how it helps crop production. So I am not an agronomist, but I think if I can collaborate with someone who has the expertise, yeah, that would be kind of cool to try it out. The only challenge is making this magnetic biochar it's a time-consuming process because I only do it in the lab. It's like pretty small. So I think we might be able to do pot experiments, but not maybe like field scale trials using this. But if there are companies that can scale up something like this, because the process in itself is not very difficult, it just requires mixing a couple of ingredients like iron-based ingredients and then pH adjustment to precipitate out the nanoparticles on the biochar surface. So it's not very, I guess, extensive. There's not a whole lot of temperature changes that's required. The process happens at about 55 to 60 degrees Celsius. So it's not a high temperature requiring process, but I don't know anyone, and I'm not an expert on like scaling things up to full scale, and I don't have any contacts who can do something like this. But yeah.
You should talk. So now, can you see the chat?
Yeah, I can see the chat, yes.
You know ICFAR?
Who?
ICFAR, it's a research institute outside, I think it's affiliated with Western in Ontario.
Okay.
So I will, because they do a ton of biochar stuff.
Oh, cool, nice.
Western University. And so here, I'm going to send this to you, and I will get you the the man's name is escaping me. But
Okay.
they do an immense amount of
I think your voice is cutting out. I can
Yeah,
barely
a little
hear
bit.
you.
There's like a threshold.
Yeah.
Sulfur is a perfectly good soil amendment. It's a micronutrient. You do need some of it. If I remember right, it tends to make things more acidic, which we don't need in a lot of soils like Vermont, but other places where there's a limestone parent material in the bedrock, yeah, then sulfur can be good. My thought on this magnetite is if it's not that expensive, especially if you can use some inexpensive feedstock like, pine chips or whatever, then you could reduce the hydrogen sulfide. It looks like the magnetite reduces hydrogen sulfide down, pretty easily down to some low-ish level, like 2, 3, 400, and then
No. The magnetite biochar did 2, 3, 400 inside the digester as an in situ process. As an external scrubbing system, 0. From 10,000 ppm, 0. There was no hydrogen sulfide in the biogas for, like I said, about, I don't know, 10 days or so.
So that's even better in a way, because my scheme is that you would use the magnetite, whether it's in the gas or the biochar, doesn't matter. I mean, in situ. So let's say you have it in situ, you come out of the digester with 300 ppm hydrogen sulfide. You could just scrub it down to 200 pretty easily with activated carbon, which is enough to meet any engine warranty.
Yes.
So 200, 150, those are engine warranty numbers. And, or, for that matter, boiler probably has a similar threshold of hydrogen sulfide. So it could be a hybrid approach on removing the hydrogen sulfide. And now you have a couple of really great, byproducts, activated carbon, magnetite with sulfur. These are both totally benign. You wouldn't have to necessarily regenerate anything.
Maybe not. Yeah. But again, it might add the cost. I don't know if you regenerate it, it might just help the farmer pay less for it. So that was my thought process. Any other questions?
It's not really a question, but thank you for the presentation. And I know in Greenland they're looking into what to do about the shrimp waste. So
yeah. So we haven't done much with shrimp waste right now, but I am. Do you know George Chamberlain?
No.
He's a big shrimp guy. I reached out. I mean, I connected with him earlier this year, and we've been talking about some biofloc stuff. Essentially using biofloc to clean out shrimp, like to kind of maintain water quality in a shrimp pond. But shrimp waste, I haven't done any work on it, but I'm really intrigued by it because shrimp is such a huge industry. And I don't know if I've seen a lot of research on anaerobic digestion of shrimp waste. But yeah, I would be really interested. Like if there is, we don't do shrimp here, but if I can get a source of waste or if I can collaborate with someone, yeah, that would be really cool.
Amber, it looks like you really want to say something.
I just get very excited. Wait, okay, so when you talk about shrimp waste, Judy, what exactly are you talking about? Are you talking about skeleton?
I think so, mainly the head and the skeleton.
Yeah.
But it probably depends on which factory, because I know some of those shrimps are sent to Thailand for deveining, and then they come back to Greenland. So I'm not sure exactly what they do, but there is definitely a lot of stuff they dump on the sea once in a while, and it smells horrible.
Yeah.
There is a company called Tidal Vision that is doing chitin-based biopolymers and bioflocs and things like that, and they're using like crab shells and the exoskeletons, the very heavily chitin.
Chitin, or flocculation.
For all kinds of things. For agricultural seed coatings, so that for all kinds of stuff. And they have they did a presentation to them on they do shrimp too. So maybe we could hook them up with your Greenlanders. I put their name in the chat.
Interestingly, I just got an email this morning because we had our compost work highlighted by USDA in their annual scientific discovery stuff. And someone who works at University of Maine Extension actually asked the same question, like, can we compost all this, seafood processing waste that includes, lobster and crab and shrimp shells? Because Maine has a lot of these seafood processing waste, and he's really interested in trying to find strategies for farmers in that state to valorize all these waste streams. So he reached out to me, and I think I'm going to have a talk with him to see if he can kind of look for funding to continue some of our composting work.
They do all kinds of stuff. They were doing, yeah.
And I think I know Tidal Vision. Why does this name sound familiar? Are they doing some AI stuff as well?
I don't think so. They seem very focused on just like, yeah, they're out of Bellingham, Alex.
Okay.
So yeah, they are. Take a look. I put their link in the.
Yep.
Yeah. Chat, but they're doing all kinds of.
Stephanie, that name, I know that name. That's someone, Alex, you connected me to, right? One time. Yeah. Grab biochar. Cool. Nice.
It sounds like something you're going to get at like a shishi Michelin-starred restaurant. Crab biochar.
Crab biochar.
Lightly toasty.
Yeah. Fun.
Yeah, check out. Judith, we can have a sideline about if this is of interest for the kind of sand.
Really exciting. Yeah. There's also a lot of fish, but they don't. For now, they don't process a lot locally, but it might happen. They are trying to develop techniques. And so the cold is not an issue, or what's the coldest you've tried with this?
Yeah. So the cold is, so we've not run our digesters at low temperatures yet, but we are thinking of doing that when we start ramping up the system. Looking at, I think we start at 25 degrees Celsius, 30 and 35. We're thinking of doing that. In our preliminary testing, we were producing gas even at 28 degrees Celsius, but we haven't been able to compare it to what would be produced at 35 or 37 versus 28. But again, we are interested in exploring that. Our other issue with cold has been like our biogas storage. We've had issues with pipes breaking and stuff like that because of ice formation and stuff. And for next year,
28, you meant Celsius or
Celsius?
Okay, just to be sure. Okay, so we're nowhere near cold yet.
No, we're not looking at those really cold temperatures. I think 25 might be our lowest, or at least that's what we're planning to test. But in the future, again, it's not part of our strategy right now, but I would be interested to see if that can be done, because the reason why that might be useful for us is because salmon production, we grow our fish at 10 to 12 degrees C. Because we have our sludge stored in a greenhouse, it usually rises up in temperature to about at least 25 degrees or 20 degrees in the winter months. And we keep the space warm as well because we have blowers and everything in there. But for a farmer who doesn't have access to any of these things, they might be interested in actually trying to digest the waste at a lower temperature. So it might be interesting for us to see that. The inoculum that I use for our saltwater research, that inoculum is actually enriched for species that can optimally produce biogas at about 30 degrees Celsius, not at 37. I haven't tested that inoculum for low temperatures, but again, because it's isolated from marine sediments, they might be more effective at lower temperature anaerobic digestion. We just don't know that yet.
Thanks.
Speaking of low temperature, I'm going to put a link in here. Embry mentioned this was the name of the paper, and Tom Curtis is, I think, or maybe the lead author on this paper. No, it's whatever. Whoever the lead author is, I'll give you the address, the DOI thing here. This goes back to 2016. And I don't think I get really skeptical. Whoops, I got an extra semicolon in there. Yeah, I get really skeptical of, having to go collect special inocula. Certainly speeds things up, but, we want to be able to operate these low temperatures in a pretty technology unsupported place. So how to do that is, I think, our big challenge. How to weatherize something enough that's cost effective and yet get some decent output even at 15 degrees C or 8 degrees C,
Yeah.
They're
also
just are patient enough to let the microbes adapt.
Yeah.
That's the kicker, right? Like we just would let the critters acclimate. I think that we get there, right?
Yeah, or we can also do what the Jaime Marti said. He just put his up in the mountains in Bolivia and just waited for the local little things to get in and do their job. Apparently, well, maybe we should just try it, put it in Greenland, and see what happens. There must be something out there that degrades organic matter, but it might take longer and we might need a bigger structure to hold everything.
Well, how much money do you have? I mean,
right
that
now,
would be
none.
none.
But,
yeah, I mean, if we could develop a budget to do something there that would use relatively simple to weatherize things, like not have to build a whole building. Those guys in Whitehorse already had a building, so that's one thing. But I don't know. I'm just thinking about it might actually be really cool because you certainly have the need in Greenland.
We could have some, I mean, if it's the fish factory, they can put it inside the factory and it's heated. But if I want to try it also for like the trail huts and the hunting huts and all the other structures out in nature. And in this case, I'm counting on nature knowing how to do the job, and we just size it well enough and then wait.
My thought process is going into even if you don't create an enriched inoculum, maybe if you mix it with like lake sediments with like an inoculum from a existing digester, I think there may be enough methanogens in those lake sediments so they can grow in that environment if you leave them long enough. Because sometimes if you get an inoculum from a different source, there may not be enough, or there may not be anything at all that can handle those low temperature systems. So even long time might not allow them to grow because methanogens grow really slow in the first place. So I think maybe having a source that is maybe acclimated to those low temperature conditions and mixing it with something that is capable of digesting organic substrates might help ramp it up, but I don't know. I'm just theorizing at this point.
There are archaeologists that study different sites around, and then they look for specific plants that show them where the soil is rich in carbon, and that would be the place where people have been throwing their food and stuff. Maybe that soil would be good because it's been collecting organic waste and decomposing.
Yeah. Something that could be a source of methanogens in those conditions might be very acclimated, or at least somewhat acclimated to those conditions in the first place. So that might help.
We just need to all move to Greenland and then we do this.
Alaska or Greenland? Yes.
Greenland. No.
Greenland, okay.
They're not going to let me move.
They won't let you move? To anywhere? Do you need help, Embry? Blink.
No. Well, I mean, I think Greenland's pretty mad at Americans.
Oh, right. Well, I'm not American, so I can probably move.
You can go.
Yeah, I can move.
And Judith, you are our in.
Yeah, I just need to. Yeah, I need to get a job again.
Okay.
I'm working on it. But I'm looking to settle down now in Sweden, I think. Sweden or Norway. And then from there, work with Greenland. But yeah.
Huh, interesting.
I can go there by train. That's nice. If you know anyone in Sweden who works on our topics, please let me know.
Merrick.
Is she in Sweden?
She's in Norway.
Oh,
Yeah.
okay. I will write Merrick then. Awesome.
Yeah, we have collaborators in Norway too, but I don't know anyone who's working in anaerobic digestion. But I know someone who at DTU, Denmark, who works on anaerobic digestion of marine wastes. And he might know people in Norway.
Who is that?
Carlos at DTU.
Just Carlos.
Well, Carlos Letelier Gordo.
Okay. I've seen him somewhere.
Robin Smith.
You might have.
Robin Smith hasn't been coming back for doing, but he did a lot of work for large digestive projects in Finland.
Okay.
And might have contacts in the other Nordic countries as well. I think he knows the big fellas, right?
The sound is not very good from. Sorry.
I think you need a new microphone, Embry.
Do you want to type it?
Zoom.
But thanks for presenting. So I missed the beginning, the introductions. Where are you based, Abhinav?
So I am in Shepherdstown, West Virginia. I work for a company, a nonprofit company called Conservation Fund.
Okay.
And my institute is called Freshwater Institute. We are basically a subset of Conservation Fund. And our area of expertise is recirculating aquaculture systems. And my job is essentially to valorize the wastes coming out of recirculating aquaculture systems.
Okay.
So that's what I've been working on for the last 6 years. Anaerobic digestion, composting, and we just started some microalgae work recently.
Mm-hmm. Okay. And mostly to be using outside of the aquaculture, or do you want to reuse on site?
So mostly to use it outside, but with microalgae, there may be potential to reuse the water. But we're not there yet. We're still very early stages. But for now, it's just external, just a way to help farmers bring in more revenue from their waste instead of just throwing it away.
There's a lot happening on that also in Norway, obviously.
Yeah. But Norway does their own thing because we have collaborators there, but they're much bigger than us, like Sintef and Nofima. We follow their work and we have contacts there, but they have a lot more, I guess, funding for some of this stuff. I think we are one of the few groups in the US doing this work with aquaculture waste. And I'm slowly starting to see more people getting into it. But we started like 6 years ago, and I don't think at that point of time anyone was doing anaerobic digestion or waste to value studies in the US. But now, because there's a lot more investment in recirculating aquaculture systems in the US, there's been a push by the USDA to kind of do more research so that they can help farmers.
Eat the algae.
Do you want to meet on Teams? We can go on Teams.
There's my little spiel on biochar and algae and all that stuff. Just been thinking about what we've been talking about.
Yep.
Yeah.
Feeding the algae to the fish is an option, but the regulations will just stop us from doing all of it. I know about Grossman Technologies.
Yes.
They actually, Purdue actually used their system for their algae studies with tilapia waste.
Yep. And then it knocks apparently, like, you can separate via algae about half of the nitrogen, half of the phosphorus, and it only works if you do both, really. So it doesn't do one or the other because it needs both nutrients to thrive.
Yep. Yeah, we'll have some results hopefully next year when we start finishing up some of our lab scale studies with microalgae. Piloted wastewater effluent polishing. Nice. Which strain are you using? Which species are you using? Chlorella? I have to convince you. Yeah. Anything else?